Hello everyone, my name is Eirini Tarsani and I'm a research fellow at Roslin. I'm working with Andreas Kranis for the work package 4.6 for the GENE-SWitCH. And today we will discuss about the Genome-wide association studies. Specifically, we conducted here GWAS for the trait of body weight in broilers using the SNP chip data and sequence data respectively. The goal of this presentation is to compare the SNP signals between the GWAS using the SNP and whole genome sequencing data. Our data consisted of 60,000 animals. We used the same animals for all the GWAS analysis. And for the GWAS using the SNP data, we used the 50K SNP chip. And we first performed, we applied the quality control criteria. After the QC filters, a number of 57K samples and 45K autosomal markers were retained for the further analysis. For the GWAS using the sequence data, we had 13 million variants and the SNP genotypes were imputed to the sequence data. For the genotype imputation, we used the Beagle software. For the statistical analysis, we conducted the same linear mixed model as described here, and we used the GCTA software. Note here that we estimated the genomic relationship matrix, and as a genome-wide significant marker was regarded here this one with FDR p-value lower than 5%. Finally, the significant markers were annotated using the ensemble web tool to predict the consequence type of the variants for the species. Our results for the GWAS using the SNP data revealed a total number of 11 genome-wide significant SNPs across 2 chromosomes. As you can see here in the Manhattan plot, the 9 significant SNPs were detected on chromosome 2, while the rest of them were detected on chromosome 11. These markers are highlighted with green color. This table presents the results by web for these markers, and we observe that almost all of them were intronic variants and were lied within genes. For example, the first marker was an intronic variant on chromosome 2, and it was detected within the GPNMB gene, which is a glycoprotein gene that has been reported to regulate the abdominal fat deposition in chickens. And we found 270 sequence variants within that gene. Comparing the results between the GWAS using the SNP and sequence data, we found that there was a concordance. Specifically, there were 3 common markers between the GWAS. As you can see here, they are with the red color. 2 of them were lied on chromosome 11 and 1 on chromosome 2. Here you can see the regional association plots, and there are some nice peaks. Although these markers were intronic variants, were surrounded by coding sequence variants. However, the GWAS using the sequence data highlighted more significant variants. For example, you can see here the plots per chromosome for chromosome 8, 19, 20, and 23. There were 243 significant SNPs for chromosome 8. To conclude, we observed the additional GWAS signals when using the sequencing data. So our findings suggest an improvement in power and precision when using whole genome sequencing data. However, the use of sequencing data is still expensive and time-consuming. We should also consider that the genetic architecture of body weight is polygenic, and under the assumption of the polygenicity, a large number of genes with small effects is expected to control the trait. So the final question is how we can use our results for the genomic prediction. We can see the proportion of the genetic variants that the significant markers explain, or even build a weighted G matrix for the genomic prediction models. I would like to thank you for your attention and thank Aviagen for providing the data.